Welcome to today's webinar, Rapid Sterility Testing, an Alternative Microbiological Method webinar is presented by Andrew Taylor, Microbiology Lab Supervisor for ARL Biopharma. Andrew has been with ARL since 2014. Prior to joining the organization, he worked as a research microbiologist at the Center for Disease Control and Prevention in Atlanta, Georgia. Andrew received his Bachelor of Science in Zoology, Biomedical Sciences from the University of Oklahoma and a Master of Science in Forensic Sciences from Oklahoma State University. Today's webinar is accredited through ACPE and is worth 1 contact hour. CE credit is applied following completion of an evaluation link found on the last slide in the presentation handout. You can find the handout on the lower portion of the GoToWebinar toolbox under Handouts. We are recording this session and will include the recording and evaluation link in a follow-up email. Questions may be submitted throughout the webinar using the GoToWebinar toolbox on the right side of your screen under the Questions tab. We will answer the questions submitted following the presentation. If we do not get to your question today, a list of questions and answers will be emailed to all attendees following the webinar. Thank you for joining today's session.
Okay, thank you very much, Amy. Again, the title of today's webinar is Rapid Sterility Testing, an Alternative Method for Sterility. Again, I'm Andrew Taylor, Microbiology Lab Supervisor at ARL Biopharma. The purpose of this webinar is purely educational, where I'm declaring no conflict of interest. As Amy mentioned, this webinar has been approved for 1 hour of CE credit. And this slide includes additional information for the CE credit, including the ACPE course number. So to get started, we have 5 goals for today's webinar, and those are to define rapid sterility methods, to compare and describe some of the most commonly used rapid sterility methods, to explain rapid sterility method validation, and to summarize what multiple regulatory bodies, which includes the FDA, USP, PDA, and state boards, have to say about the matter, to outline factors that a firm should take into account when implementing the use of rapid sterility testing, and finally to highlight several of the benefits that rapid sterility testing offers. So rapid sterility methods are part of a larger group of tests termed rapid microbiological methods, or RMMs. This term generally includes rapid sterility test methods, as well as being a little bit more broad in including bioburden testing, rapid environmental monitoring, and so on. Of course, for this webinar, I'm going to focus on the RMMs that are used primarily for sterility testing. So, what is rapid sterility testing? These are tests which offer a faster alternative to traditional sterility testing, such as USP 71. Traditional culture-based sterility testing techniques require extended incubation times, which then take a relatively long time to produce final results. Since timely microbiological data are often critical, not only for product release, but also for continuous process monitoring and control, rapid sterility testing is becoming more prevalent and more useful. In order to reduce and in some cases eliminate the incubation times required, rapid sterility systems use modern technology and automation to detect the presence of microbial contamination. USP 797 states for products that require sterility testing, the testing must be performed according to USP 71 or a validated alternative method that is non-inferior to that USP 71 testing. Many rapid microbiological method technologies have been developed over the last 20 or 30 years. Most of these really have been marketed heavily in the clinical sector. However, some of those have specific pharmaceutical applications, where others have actually been developed solely for the pharmaceutical industry. Here is a list of various rapid microbiological methods, and that is organized by type of determination, detection method, and so on. Today I'm going to go into a bit more detail regarding some of the most common rapid microbiological methods, which are used as an alternative to traditional sterility testing. And those are ATP bioluminescence, CO2 detection, and solid phase cytometry. As you can see, these three methodologies vary with respect to detection method, and importantly as well as time to result. I'll cover both of those in a bit more detail later in the presentation. Growth-based rapid sterility technologies are often started in the same manner as conventional sterility testing, but then they differ in that they rely on the detection of either biochemical or physiological growth indicators rather than that typical visible or macroscopic growth. This generally allows for a much more rapid detection compared to traditional methods, but it does require that variable enrichment or growth stage before microorganisms can be detected. That is especially true in samples which contain low levels of contamination. The first type of growth-based rapid sterility testing methodology I'll be discussing are those based on ATP bioluminescence. Adenosine triphosphate, or ATP bioluminescence, is a well-established rapid method for assessing contamination levels in pharmaceutical products and in raw materials. Specifically, this method utilizes a substrate and enzyme combination, that's luciferase and luciferin, to break down microbial ATP from growing cells to then produce visible light. This visible light can then be measured using a luminometer, where the amount of light measured is relative to the number of microbial cells present. One of the main drawbacks with this technique is that care really must be taken to minimize the presence of non-microbial ATP in a sample or during a test process, as that also can be detected and generate false positive results. The following slides include examples of systems which employ ATP bioluminescence technology. One commonly used rapid sterility system which utilizes ATP bioluminescence is the Milliflex Rapid platform from EMD Millipore. Testing using this system entails the filtration of a sample, where then that filter is placed on an appropriate agar medium to allow for growth of microbial colonies if they're present. These colonies are then treated with an ATP releasing agent, followed then by the addition of that luciferin and luciferase combination, where photons of light from each colony are then detected by the luminometer and a cell count is reported. With this technique, it is also possible to continue incubation to form larger colonies or more colonies in the case that subsequent microbial identification is necessary. Some of the known strengths and weaknesses regarding the Milliflex Rapid system are listed on the table on this slide. As is the case with multiple methods I'll be discussing today, one of the biggest strengths with this system is that the results are instrument generated, meaning they are objective and require no analyst interpretation. This system relies on the growth of microorganisms, which is both considered a strength and a weakness, in that it is more akin to traditional sterility testing, but the growth step does require additional time prior to results generation. Since this system does utilize a membrane filter, that may limit the types of samples which can be tested to only those that are filterable, though depending on the product type, it may be possible to perform pre-test processing in order to make certain samples filterable. As mentioned on the previous slide, this method does actually allow for microbial identification in the event that an investigation is required. So the fact that the contaminant is not destroyed or lost is definitely considered a strength for the Milliflex Rapid system. A second common rapid sterility system which utilizes ATP bioluminescence is the Celsus Advanced platform, and that's produced by Charles River Laboratories. Testing using the Celsus system is actually initiated exactly like a traditional sterility test, where a product is first processed using a suitable method, and that can be either membrane filtration or direct inoculation. The sterility test media are then incubated for an appropriate amount of time, That appropriate amount of time is based on the method validation, which we'll discuss later in the presentation. After that incubation period, aliquots of both media are placed into small cuvettes and then loaded into the instrument. The Celsis Advanced system then adds the luciferin and luciferase enzyme combination, which allows for the conversion of any microbial ATP present into light. One notable benefit about this system is that it actually allows for full walk-away automation. So an analyst can load the samples, start the assay, and then go tend to other business while the assay is running. Here are some of the other recognized benefits as well as weaknesses associated with the Celsis Advanced system. Similar to the previous systems discussed, the results produced by the Celsis Advanced are instrument generated. The elimination of an analyst or analyst interpretation is considered very beneficial with respect to data integrity as well as consistency in methodology. As with all growth-based systems, testing performed using the Celsis Advanced is built on the premise of traditional sterility testing. But again, that enrichment or growth stage does add time to the assay before a final result can be generated. Unlike methods which require a sample to be filterable, one notable strength associated with the Celsis Advanced system is that it allows for great flexibility in sample types. Suspensions, oils, and really any sample which can be tested using traditional methodologies can also be tested using the Celsis Advanced system. Importantly, adequate suitability testing must be performed to ensure that non-microbial ATP is absent from the sample, and that the sample properties themselves don't slow or inhibit the reaction that this technology uses. At this point, I'd like to quickly remind everyone to ask any questions you may have using the webinar control panel. We'll be answering many of those at the conclusion of this presentation. A second type of growth-based rapid sterility methodology that I'll be discussing is that of colorimetric growth detection. So this detection method relies on a color change being produced either in test media or other assay materials as a result of microbial metabolism during their growth. Often this really applies specifically to CO2 production. This can limit the range of microorganisms that can be detected by this method based on their specific metabolisms. Systems are currently available which are able to detect most aerobic organisms as well as certain acid-producing microorganisms such as lactobacilli and yeasts. One common rapid sterility system which utilizes the colorimetric detection is the BACT-ALERT 3D, and that's from bioMérieux. This system was originally designed for clinical applications, but has since been found to be suitable for some pharmaceutical testing. Testing using the BACT-ALERT 3D equipment begins with samples being added directly to bottles of liquid culture media, which are then incubated inside the system, and that's at one of two different temperatures. During certain types of microbial growth, CO2 in the closed container would then accumulate and diffuse into a sensor at the base of the bottle, which then produces a measurable output. One notable benefit of the system is that it automatically monitors the sensors, and it does that every 10 minutes, so that provides very up-to-date test data. On this slide, we have a list of strengths and weaknesses associated with the BACT-ALERT 3D. Again, like the previous growth-based systems mentioned, the results produced by this equipment are instrument generated, eliminating that subjectivity associated with manual reads. This system is unique compared to the others in that it offers continual monitoring. Again, like I mentioned, reads every 10 minutes, which can be extremely beneficial with respect to real-time results. This allows for a much more timely approach to any out-of-specification investigation or other potential contamination events. A notable drawback of this system and other similar systems, like I mentioned, is that the detection can be dependent on the microbial contaminant's specific metabolism. This may limit the range of microorganisms which can be detected using this technology. And the last category of rapid sterility methods I'm going to discuss today are those which are non-growth based. These systems don't rely on microbial growth to detect contamination, but instead they rely on cell labeling techniques or microscopy to detect and quantify viable microorganisms. This approach really has the potential to detect a wide range of microorganisms within only a few minutes or hours. With that said, some systems do require an enrichment or growth step if very low levels of contamination are present or are suspected. That can be levels of contamination around less than 10 CFU per mil or lower. One of the most well-known examples of a non-growth-based rapid sterility platform is the Scan RDI. That's also by bioMérieux. With this system, the test sample is filtered through a membrane where any microorganisms that are present are retained on a filter. During the assay, a fluorochrome is released and then retained inside the cells with intact membranes. The equipment then scans the surface of the filter within approximately 3 minutes, where any viable cells are detected. Based on certain system settings, autofluorescing particles, membrane fluorescence, or other background noise are rejected, and a total viable count is reported. It's noteworthy that many firms also include a step where positive results are manually observed using a microscope, and that's done for confirmation of contamination. The biggest strengths associated with the Scan RDI system really is the combination of speed and sensitivity, where detection down to a single bacterial or fungal cell can be performed in a matter of minutes or hours. When time to results is a high priority, any system which does not require a growth step offers notable benefit. As with other systems which utilize a membrane filter as part of their method, the sample types which can be tested are thus potentially limited to those that are filterable. And again, as I mentioned a moment ago, there may be sample processing options that make certain products filterable. Regarding results generation, because there is often an observation step to confirm contamination with the Scan RDI, results are somewhat more subjective compared to many growth-based systems which do not require that analyst involvement. And finally, since no growth stage is afforded, microorganisms which may have been present in the test sample are actually lost during testing. This means that should contamination not be homogeneously distributed in a product batch, it may be impossible to fully investigate or identify those microbial contaminants. So moving on to the next portion of today's webinar, perhaps the most important facet of employing a rapid sterility method is confirmation that a thorough method validation protocol has been completed. All of the systems mentioned during the previous slides have undergone full validations by their respective vendors, as well as by numerous labs around the world. Since this validation is considered hugely important, but that they can vary based on the technology used, as well as different labs' implementation, the FDA has not yet blanketly accepted any rapid technology as a whole. They are, however, currently open to reviewing validations of methods where they've been approved on a case-by-case basis. To address one question that gets often asked to us here at ARL, and that is, does my state board of pharmacy allow for rapid sterility testing? Currently, most state boards do allow it, and we recommend that you check with your state board directly for more information. Historically, the task of implementing rapid sterility testing was considered very difficult and actually a barrier to entry for QC microbiology labs not familiar with those alternative rapid methods. However, regulatory agencies have since responded and have published additional guidance, as well as documentation to assist in that implementation. Within the United States, PDA Technical Report 33, as well as USP 1223, can both serve as great starting points for the validation of any rapid microbiological methods. USP 1223, which is titled Validation of Alternative Microbiological Methods, is actually directly referenced in USP 797, which states, again, sterility testing must be performed according to 71 or a validated method. The subsequent slides delve more into the parameters which must be assessed as part of a validation protocol based on the guidance from USP 1223 and PDA Technical Report 33. This slide shows Table 1 from USP 1223, which is titled The Validation Parameters by Type of Microbiological Test. As you can see, the table is broken down based on quantitative and qualitative test methodologies. Since sterility testing is a qualitative test, meaning that it yields a result of sterile or not sterile, we'll focus on the left column. The specificity of an alternate qualitative sterility method is defined as its ability to detect a range of challenge microorganisms specific to that technology. A range of microorganisms can be defined several ways, but at minimum, validation of a rapid sterility method should include the 6 microorganisms listed in USP 71, and oftentimes it will include the addition of various other environmental isolates, organisms identified from previous product failures, representative slow-growing organisms, and so on. Specificity is demonstrated by using an organism challenge level that is above the limit of detection, and I'll talk about the LOD on the subsequent slides, generally around 100 CFU of each microorganism, where both the compendial and alternative methods are performed with the expectation that comparable recovery is seen. The limit of detection, or LOD, of a technology is defined as the lowest number of microorganisms that can be detected under the stated experimental conditions. To determine the LOD, serial dilutions are made, which include a range of concentrations of each challenge microorganism from the panel I previously mentioned. The level of inoculation should be targeted or adjusted to target only about 50% of all samples showing growth using the compendial test. Commonly used CFU counts are approximately 5 CFU per test, approximately 0.5 CFU per test, and approximately 0.05 CFU per test. Again, this is done with the expectation that negative results will be observed using both test methods. Both the compendial and alternative tests are performed using a sufficient number of replicates to allow for appropriate statistical analysis. Oftentimes that's the chi-square test or other similar analyses. Repeatability is defined as the degree of agreement among individual test results when the procedure is applied repeatedly to multiple samplings of the same suspension of microorganisms. This is done primarily to ensure that the results are both consistent and repeatable. Robustness is defined as the capacity of a method to remain unaffected by small but deliberate variations in method parameters. Those method parameters can be reagent volume, incubation time, ambient temperature, and so on. Slight variation to those parameters provide an indication of the reliability of the method, as well as the equipment during normal usage. It's important to understand that the measure of robustness is not a comparison between the compendial and the alternative methods. Rather, it's a necessary component of the validation of the alternate test so that the user truly understands the limits of the operating parameters within that method. Ruggedness is defined as the degree of precision of test results obtained by the analysis of the same samples under a variety of typical test conditions. Those test conditions can be things like different analysts, different instruments, and different reagent lots. The expectation is that test results obtained by testing the same sample with differing or varying analysts, instruments, or reagent lots should yield consistent results. It is noteworthy that it's not uncommon for the end user to rely on data supplied by the test system manufacturer for both robustness and ruggedness determinations. When validating rapid methods for sterility testing, including stressed or injured microorganisms is highly recommended. Those types of microorganisms are much more representative of potential contaminants that may be present in pharmaceutical samples to be tested. PDA Technical Report number 33 specifically mentions that a complete method validation should include the successful detection of those stressed microorganisms. In order to perform this testing, methods are often used to stress or injure different test colonies or cultures. Some of those methods are or can be to expose microorganisms to certain environmental conditions, such as UV, heat, cold, pH adjustments, or to certain antimicrobial conditions, so exposing them to disinfectants, or to put them through certain sublethal sterilization conditions. Either concurrently or prior to testing, these stressed microorganism cultures should be plated and enumerated to ensure that the appropriate reduction in viability is seen. The expectation is that reduced CFUs would be present in the injured group compared to the control or healthy cultures being used. Testing is then carried out similar to how it was for previous validation tests, in that both the alternative and traditional methods are performed using a sufficient number of replicates to allow for appropriate analysis. Ultimately, the aim of all this validation protocol testing is to demonstrate the acceptability of the alternate procedure relative to the current microbiological practice. To do that, the laboratory must demonstrate that the new procedure is as good as or better than the current procedure in terms of the ability to detect the presence of microorganisms. This is determined to be the equivalence. USP states that alternate methods may be used if they provide advantages in terms of accuracy, sensitivity, precision, selectivity, or adaptability to automation. It also says that they must be shown to produce non-inferior results compared to the compendial method. Similarly, FDA guidance states that a validated alternative procedure must be shown to have performance equal to or better than the traditional procedure. When comparing two test procedures to show equivalence, statistical evidence is assembled to demonstrate that non-inferiority. For example, with sterility testing, equivalency is shown if there is no statistically significant difference between the two groups of results generated when performing testing with the compendial and the alternative methods. At this point, I would like to once again remind everyone to enter any questions you may have using the webinar control panel. The next slides cover some important things to consider when deciding if rapid sterility testing is right for you. First and foremost, it's always important to remember that patient safety is best served by detecting any microbial contamination prior to a product's administration. Ultimately, in order to determine if a rapid sterility test is right for you, and further what methodology meets your needs, several factors play into that decision. This includes time to results. As mentioned previously, methods which do not require a growth step offer the fastest turnaround time. If time to result is the most important factor for you, looking into one of these may be your best option. Second is the specificity and sensitivity of the method. Can it detect the appropriate range of microorganisms such that you feel confident releasing a product based on a sterile result? Next is sample size. Do you or does your testing lab have different sample size requirements? Often this is still based on guidance from USP 71 in tables 2 and 3, or direction from USP 797 for smaller batch sizes. It's also possible that labs have developed a justification for modified sampling requirements. One other point to consider would be your product attributes. For example, many compounded sterile products or cell therapy products, for example, due to their short beyond use dating, BUD, would benefit from an overnight test or at least one that's completed within 48 hours, where other products may not require such a short time to result. Other factors to consider when deciding which rapid sterility test is right for you is the ability of the system to support aseptic material handling. What that really means is, is the system closed? Does it inherently reduce contamination based on its design? Or perhaps does it do the exact opposite, where increased false positive results might be seen? Next, if you're using your own lab or a contract testing lab, as with any analytical testing, it's important that you have confidence in the knowledge and the experience of the group actually running the assay. As mentioned during previous slides, various methods allow or limit the types of samples to be tested, filterability as an example. So you might decide if, based on your production specifics, does this impact you or your ability to achieve rapid sterility results for your entire range of products? Lastly, I would like to note, just like with any thorough sterility test, method suitability testing is required to ensure a proper method is chosen for each unique product formulation. Sampling requirements for this method suitability will vary based on methodology and a laboratory's requirements. However, it's important to note that this is definitely a needed step prior to performing rapid release sterility testing. Rapid sterility testing offers numerous benefits compared to a traditional sterility test. Most notably, of course, is shortened test times. The wait for a traditional sterility test result introduces additional storage requirements, merchandise hold times, delays to market, as well as risk into the production process in the event of a contamination event. As noted previously, certain products with short shelf lives are prime candidates for testing using rapid sterility methods. Faster test times allow for improved efficiency across many facets of a production cycle, which includes storage and distribution. This testing can also result in reduced waste due to products exceeding their BUD while sitting on shelves or in a warehouse somewhere. These efficiency gains also create further benefits by allowing firms to more rapidly respond to their patients' or their clients' needs or requests. In addition to gains in efficiency and production, an extremely noteworthy benefit to shortened sterility test times is that it offers the ability to perform investigations in a more timely manner. Rather than waiting 14 to 18 days for a traditional sterility test result, being given that under investigation or not sterile result closer to the date of production allows for a much more effective investigation. It also allows for the ability to potentially implement any appropriate corrective actions much sooner. So with a faster response, then comes a faster recovery following any contamination event. The benefits of rapid microbial detection extend beyond just faster time to result. While the faster result is obviously a major positive impact of rapid detection, users of many systems also gain the ability to address and better meet data integrity expectations and requirements from their governing agencies. With some of the technologies discussed in this presentation, the implementation of a rapid sterility method replaces the subjectivity of traditional visual confirmation with an automated assay controlled by an instrument. Results are controlled and reported through the software, thus avoiding transcription and interpretation errors potentially caused by an analyst recording or reporting handwritten results. Instrument use logs can also be generated, which can help meet certain audit trail requirements. The picture I have on this slide shows 4 separate sterility test media broths from a traditional sterility test, each demonstrating a different visual appearance. In this specific case, the 2 samples on the left are sterile, and the 2 on the right are contaminated. While a traditional sterility test method is able to overcome this issue by subculturing any turbid or questionable material or media, this does highlight the fact that the use of only visual observation may lead to inaccurate or at the very least delayed results. So in summary, rapid sterility testing is a group of methods which are part of a broader group termed rapid microbiological methods, or RMMs. Rapid sterility methods are alternate test methods to use in place of or in conjunction with traditional sterility testing, USP 71 as an example, which allow for shortened or in some cases no incubation times. These methods often used advanced technology to detect microbial contamination, which allows for faster test times since the user does not have to wait for visible growth to appear. Numerous vendors have created rapid sterility testing systems which employ different modes of detection in order to provide an alternative to traditional sterility testing. These systems differ with respect to testing time, meaning whether there's a growth or enrichment stage, product types that are able to be tested, example would be filterables versus non-filterables, subjectivity of results, are they instrument derived or does it require an analyst's input? And finally, the ability to further investigate out-of-specification results. Is the microorganism able to be identified once contamination is suspected? These are all factors to take into account when deciding which rapid sterility test method best meets your needs. Rapid sterility method validation is a multi-step process with the ultimate goal of demonstrating equivalence, or in some cases superiority, of an alternative test method to a compendial test method. As noted, there are multiple resources available to assist in the design of a validation strategy, with notable recommendations and guidelines being produced in USP general chapters, FDA guidance to industry, and PDA technical reports. One final important note to reiterate is that method validation is completely separate from formulation-specific method suitability. Once a rapid sterility test method has been validated by a lab that plans to use it, each formulation to be tested still requires method suitability to be performed in order to demonstrate its compatibility with that system. The use of a rapid sterility test is of course, a risk-based decision where the stakeholders must bear in mind that patient safety is always paramount. Good manufacturing practices and a superior quality system management must be maintained, irrespective of whether you choose to move forward with a rapid sterility method or not. Multiple factors, including time to result, specificity and sensitivity, product applications, sampling requirements, as well as the knowledge and experience of the testing lab, should be taken into account when considering the implementation of rapid sterility testing. Rapid sterility testing offers multiple advantages compared to traditional sterility testing. Most notably are those shortened test times. These create benefits across an entire production process, including the release of products faster. With a faster test time also comes the ability to perform investigations and implement corrective actions in a much more timely manner. And additionally, reduced subjectivity in results, as shown in some of the examples mentioned earlier, as well as improvements to data integrity compliance are both benefits associated with moving to rapid sterility test method. And as a final summary, time is money. A faster test time allows you to release products faster, reduce inventory and improve warehouse utilization, shorten production cycles, respond to and recover from contamination faster, and overall improve efficiency. So here is a list of references that I compiled in preparation for this webinar. And we will now transition to the Q&A portion of the presentation, where we will address any questions that have been asked. Thank you very much for your time today.
Thank you, Andrew, for that fantastic session. We will now begin our Q&A portion. As a reminder, please submit questions using the GoToWebinar toolbox on the right side of your screen under the Questions tab.
Okay, first question here I see is, why is including stressed organisms important during the validation? So I sort of alluded to it in the presentation, but stressed or injured microorganisms are much more representative of what one would expect to find in a compounded pharmaceutical product. As you can imagine, when organisms may be present in such an environment, they are not happily growing. They are not in any sort of proliferative stage. So, and they're often in very small numbers. So including things other than microorganisms in their ideal state during that validation is considered extremely important, which is obviously why it's referenced in PDA Technical Report 33.
And as a reminder to all of the attendees, we also have Brian Kelly on the line here, Director of Business Development, to answer questions along with Andrew.
There's a question here. How does the FDA view rapid sterility methods utilized in CGMP environments in lieu of USP 71? I can speak to the first part of that. Brian may have a little bit of additional information. As I mentioned in the presentation, the FDA is open to reviewing and approving method validation protocols using rapid sterility testing techniques. They just haven't blanketly accepted any one technique over the others that are available. I think the primary reason for that, again, is the differences that labs or different product formulations may have when they're interacting with different methods. Brian, do you have any more thoughts?
Yeah, nothing else to add. Andrew hit on it there. It's not really the technology itself that's approved by the FDA. It's the combination of the technology with the validation. So they'll want to review both of those things before they agree that the necessary validation has worked and that non-inferiority was proven through the validation. Andrew also mentioned the method suitability, so that's the formulation-specific validation, right? So you have the validation of the technology or the methodology as a whole, but then there's that formulation-specific piece, which is the method suitability that also has to be picked up.
Okay, and then another question here, sort of related. Is FDA okay with rapid sterility testing? I think we answered that. Would they write issues related to this testing on a 483? And I think that really goes back to making sure that an appropriate validation protocol has been performed and is available. One could easily imagine information about this getting put onto a 483 if the appropriate validation work hadn't been done, or again, if certain methods had not been demonstrated to be acceptable for the formulations that are being tested. Here is another question. What rapid sterility system does ARL use and why? We use the Celsius Advanced platform. One of the primary reasons we chose that platform when we were assessing different options was primarily twofold from our perspective. One, it's closely in line, or it is based on traditional sterility testing methods, which are growth-based, so that's much more akin to what we're familiar with and what we've utilized for years. But I think the most important thing for us was that it's a non-destructive test, and that is if you can, or if you get a non-sterile result or a result that's under investigation, you can investigate that fully, and you can ultimately identify that contaminant which is present, and that is absolutely the expectation with many regulatory agencies when you have a contamination event. You good, Brian? Yeah. Why does media sometimes become turbid without contamination? Oftentimes those are what we call sample properties. So when you're testing maybe an oil or a suspension that doesn't fully filter through a membrane filter, part of that sample can remain in the test sample, which can appear as either turbidity, cloudiness, again, oil that settles towards the top, which may make a visual read difficult. So in traditional sterility testing, you would subculture that into fresh media and observe for potential growth after additional incubation. Whereas a lot of the rapid microbiological methods, because they're not using visual turbidity as the indication of contamination, can actually bypass that step and give you either a more accurate result or at least a more timely one. You mentioned in the presentation that the initial test setup using the Charles River system mirrors USP 71 testing. Can you go into more detail about what that means? Yeah, and I alluded to that in the answer I said a moment ago about which system we use, and that is that it is, in using the Celsus Advanced system, the test setup is exactly like a 71 test setup, which is if a formulation requires membrane filtration, we can use membrane filtration, set that test up, put it in the incubator, and begin incubation. Or if it's a sample that's not filterable, we can perform direct inoculation, just like we would with the 71 test, put those samples in the incubator, and then test that media using the Celsus luminometer after the incubation period. So that comfortability with USP 71 and how it's set up was one of the reasons, again, we chose the Celsus Advanced platform. And the diversity in sample types is another important factor that we took into account in wanting to use that system. The example sample tubes you showed, number 3 was clear, and you said it was contaminated. These are not visual tests. Perhaps that was just not clear in the PowerPoint presentation, but there was some slight turbidity in that, and some microorganisms' growth patterns can find their way down to the bottom of the filter, such that you can't see that in a lot of the growth media unless you swirl it and or do other kind of manipulation of the test media.
But I think kind of the point is, so what Andrew was trying to show there was that the traditional 71 test, the results are obtained by visual observation, right?
Correct.
But with these alternative sterility test methods, they may actually pick up on a contaminant that would be hard to detect visually. Right?
Sure. Yeah, that may be difficult to see, maybe in very low quantities. We've seen that with certain organisms and their growth characteristics. Very slow-growing organisms may not actually create turbid sterility media until very late in the incubation period, or even after the 14 days. So oftentimes the sensitivity with the rapid methodologies is hugely important there too. We have can emulsion or suspension be done through rapid testing? So again, that's going to depend on the platform being used. Certain platforms require a product to either be filterable or be amenable to certain processing techniques, which then make them filterable. And depending on the formulation specifics of the emulsion or suspension, that might not be possible. So that may limit certain emulsions, suspensions, or other sample types from being tested on certain rapid platforms. Another question here, it's a good question. With the shortened time to test results, does the out-of-specification investigation also shorten, or does it sort of revert to the regular process? So as I mentioned in the presentation, one of the big benefits with rapid testing, or really getting results quicker, is getting an under investigation or a not sterile result quicker just shortens the overall process for the entire investigation. From the lab side, we are closer to the date of test. We are closer to collection of the data surrounding the processing of that test, looking for errors and so on. And then from the client's perspective, you're much closer to when that sample was compounded. So you can more easily access your batch records, your quality records, and get that investigation initiated much faster. So take a rapid sterility test results that is released after 5 days compared to one a traditional sterility test that's results are generated after 14 days. You're already more than a week ahead where you would be with that traditional sterility test. Here's one. What could cause a false positive result with the Celsus Advanced platform? So as I mentioned in the slide with the table on the strengths and weaknesses of that platform, it can detect non-microbial ATP because it's detecting ATP, right? So in certain biologically derived products that contain, whether they're either cell-based or serum-based products, those might actually generate an elevated result because ATP or an ATP equivalent is present, even though it's not from microbial contamination. Charles River does offer there are processing techniques which can either minimize that or get rid of it completely, and that can just vary based on the specifics of the sample to be tested.
I think some of that also goes back to the original test setup, right? Because just like with the traditional USP 71 test, contamination can happen during the test procedure itself, right? Which is why you want to make sure that the laboratory is minimizing the chances that's going to happen by like using closed membrane filtration systems and things, right? Things that are going to prevent the contamination during the test, because as Andrew just said, the system doesn't have the ability to distinguish between microbial ATP and ATP for some other source, right? But the microbial ATP, the source of it, could come from the test environment, the testing personnel, just like it came from the compounding environment or compounding personnel. So there's steps to do that can mitigate those risks, but that possibility does exist.
So do these test for endotoxins? No, but endotoxin test methodologies are available, which can generate results in just as fast of a turnaround time. So whether you're bringing these technologies, into your facility or you're working with a contract testing lab, you can work with them on different turnaround times or expectations for testing for endotoxins to kind of go along with what you expect with your sterility test turnaround time. Does ARL offer non-growth-based options like scan RDI or only the Celsis? At current, we only offer the Celsis rapid sterility test methodology, and that's primarily for the reasons I mentioned earlier. You said that the FDA does a case-by-case basis for rapid sterility, but do you know how boards of pharmacy and surveyors like the Joint Commission see rapid sterility testing?
Yes, I can't speak so much to the Joint Commission, but as far as state boards of pharmacy go, we're only aware of one that has not accepted any rapid sterility methods under any case, and that's California Board of Pharmacy currently.
Here's one that says, how do you determine which testing method works best? My question is because we know that different organisms may be easily found using one particular method versus another one. And really that's going to go to the risk-based decision you need to make in determining which method you employ, whether, again, it's at your facility or a contract testing lab. If certain technologies do not detect all microorganisms that you consider required to be detected. So again, at minimum, that's going to be the USP 71, 6 microorganisms. Again, oftentimes slow growers, it's very important to include those in validation protocols that certain rapid techniques may not find. Ultimately, it's really going to be very case by case to my mind. I think working with or asking your contract testing lab what their validation protocol looks like, making sure you're comfortable with it, perhaps getting a copy of it and blessing that or asking any questions. Those are all sort of important questions or things to do as you're assessing whether rapid technology or rapid sterility makes sense for you and your firm.
And you mentioned that one technology by Byron Maru that isn't capable of detecting all types of microorganisms, right?
Yeah, that was the BACT alert, which is based on CO2 production. So not all microbial organisms produce CO2 in their metabolism. So in theory, those would not be found by that system. So you definitely want to take that into consideration as you're working through which testing type to use. So getting ready for the new rules, I assume that means the updated 797. Does rapid test results, will those be accepted? So again, 797, even the current version, as well as the one to be official in November, mentioned that for any product that needs to have sterility testing performed, that should be done by USP 71 or a validated alternative method. And that's where that references USP 1223, which gives direction on how to validate different methods. So again, whether you're bringing a method in-house or you are using a contract testing lab, you want to make sure the method they're using is validated in accordance with 1223 so that you're meeting the recommendations or guidelines listed in 797. And that's, again, the current version as well as the one to be official in November. Here's one. How much sample is required for these tests? So that is going to, again, depend on the different methodology to be used. I believe I alluded to it in the presentation, but to go into more specifics, what we see most commonly is people still follow the sampling guidelines in USP 71. That's tables 2 and 3. Noteworthy that the updated version of 797 to be official in November, but is available online now, does give additional clarification for small batched or small batch compounded sterile pharmaceuticals. So small batches, which would be basically less than 40, it does give a little bit more clear sampling guidelines for how to sample those types of batches. So ultimately, it's going to be a decision that the firm would need to make whether they want to sample in accordance with 797 or 71, or maybe an alternative that they've deemed appropriate. How do you handle ATP positive cultures that do not grow when subcultured? So I'm going to answer this assuming that means for the Celsius platform and assuming that means for ARL. So if we get an ATP positive culture that comes in during method suitability testing and it shows ATP presence, that's basically why you're doing method suitability testing, right? You want to determine if there's a method that you're able to overcome the inherent ATP present so that the subsequent test can measure ATP present from microbial contamination, not the sample itself. Now, if you've determined that you've found a appropriate method that gets rid of that ATP, and during subsequent release testing, you see ATP present, you would consider that a not sterile or at least an under investigation test that you can investigate for growth. Perhaps it would subculture and grow, perhaps it wouldn't. There are certain organisms that are less or maybe a little bit more finicky and difficult to grow. So you kind of have to work with your lab and make sure that an appropriate investigation ensued.
Thank you very much for Andrew and Brian for those questions in the answer session, and for all the attendees that submitted questions. If we did not get to your question today, we will follow up with an email. As a reminder, if you would like CE credit, make sure that you complete the survey on the last slide of the presentation handout to receive that. This will remain open for approximately 2 weeks. Be sure to go in there, have your NABP number, your date of birth, and you'll be able to receive the CE credit after completion of the evaluation. Thank you very much and have a great day.